All right, I guess we'll get going. I'm Ascher. I'm at the University of Arizona. I'm a PhD student. I'll be graduating in about 1 year. This is Blake. He is already a doctor, not a medical doctor. He is a biologist with a strong interest in code. I'm more of the opposite, code with a strong interest in biology. So we're going to talk about genomics and visualization, and also we're going to provide you with a nice amount of background for those who have no idea about biology and might feel overwhelmed hearing that. So let's talk about biology really fast and what is a genome? So the way I explain this to my grandma is that a genome is a set of instructions similar to a cookbook you might have, except instead of to make a cake, it's to make a living creature. Any living creature, me, you, Blake, those plants growing up on the wall. And it's a little bit different than a normal cookbook too, because it's not made up of our 27 letters that we know. It's only made up of 4, A, T, G, and C. And similar, it has some different functions that you might be relatable in code, and it's made up of parts similar to functions you'd write. They're modular. You can move them around. For example, you can see here, you can take one gene, which is one of those little pieces of a genome, out of one creature, put it in something else, and actually take that property across. So here they've taken a glowing fluorescent protein out of a jellyfish, put it in a mouse, and we have green glowing mice. You probably heard of this. It's called genetic modification. It's pretty popular in the news. I'm not saying it's good. I'm not saying it's bad. It's cool, though. And so one other important thing to note is that the genome can copy itself, and this is how life has gotten to this point. Over millions of years, it's copied itself, not perfectly though, and it's sort of changed and morphed into what we see in the world today. And so we sort of want to propose to you guys that genetics is and has always been a big data problem and a visualization problem, and it's only getting worse as we go on. So there's 3 main challenges that we sort of view in genetics, and the first is how do we actually see this sequence? So the words in your cookbook, how do we take this? Because these are small, they're not 12 font. These are tiny little chemicals that you can't see, and they're packed into your cells, and there's lots of them. So how do we see that? And then after we can see that, how do we make sense of it? So it's not organized in a nice step 1, step 2, step 3, way a normal instruction set might be. It's just all in there, and it somehow is regulating itself, and we have to figure that out without really knowing. And then the third way is just how can we actually use visualization to teach people these concepts that are fairly complicated, and they give out PhDs for it, so it's not a trivial question. So I'll start with the very first challenge here, and that's 1.1. So this is really a cool picture. This is actually what you're looking at is the very first visualization in genomics ever. This is X-ray crystallography of the genome. So the genome has this sort of double helix structure that you can see on your right, and you can see the A's match with the T's, G's match with the C's. I don't know if you can sort of see that structure in that image. What they basically did, this is essentially a shadow. They shot X-rays through the genome, and this is the image. And you can see that wrapping. Before it was thought it was just a single strand. I find it incredibly impressive that someone looked at that image and then came up with this second model on the other side, because I probably wouldn't have found that if I was the one looking at it at the time. This was done in 1952. It's a pretty recent field by Rosalind Franklin, who you might not have heard of because all of her fame was taken by 2 other men known as Watson and Crick, who you've almost certainly heard of as the founding people of genetics. So how do we read this? So your body can read your genome through the cells. It has different little mechanisms in there to copy it, but that doesn't really help us a whole lot. So this is really where our beginning to understand the genome came. So on your right again, you'll see this 100 NT. So an NT is a nucleotide. That's one of those letters. And this is how, for many years, we actually went about reading this. And it was very tedious. What they would do is they would copy it with special radio-labeled, so radioactive versions of the DNA. Then they'd chop it up and run it down a gel with the general idea that things that are bigger move slower. And so that sort of separates bigger things from smaller things. And you go down with a ruler and you count in those things, A's, T's, G's, and C's. It's a lot of work. Blake has a great story. You might want to ask him about his PhD advisor used to bring her kids into the lab and would set them there with a tape recorder and have them read these off because it was a lot of work. So then people got a little smarter. They're like, we don't like working with radiation. It's annoying, a lot of government oversight. So how can we replace this? Let's use color. So that's where the next sequencing technology came in. And of course, we had a lot of technological advancements, so it got faster. We could let the computer read it, so that was good. And we've moved on, and we're now up to some new technologies like the Pacific Biosciences, which actually takes some of those components of your cells and uses them, along with a camera, to actually read these off as it's copied. But so, while that's a really interesting technology and produces these very long sequences, this is really how we do DNA sequencing today. And this is sort of an interesting way to go about it. And we call it shotgun sequencing. And it really is like a brute force shotgun attempt. We bust it into a ton of pieces, read it off maybe 150 letters at a time, and then try to assemble it all back together using algorithms. So, big data. And this machine is actually amazing. It's so fast that you can sort of see this trend. I'm guessing you can see my mouse. Yeah, perfect. So this is basically where this came in. And you can see that the cost of a human genome went from $10 million to $1,000 in less than 10 years. So this is really fascinating. And so now we can do a human genome for about $1,000 a piece. One of these machines can do 18,000 a year. So that's one contributing growth in DNA sequencing and our big data problem. And then here's the second sequencing challenge. We have a lot of them. They're all around the world. Every single continent has a number of these sequencers. And these things are running around the clock. I mean, they're expensive machines, and you don't turn them off when you go to bed at night. You let them run. So you can sort of see time. We're getting a lot of data. We have billions of nucleotides here. That's a 200 at the top. It's overlapped with my labels. And over 200 million different entries into these publicly available databases. So this is data I could go download onto my computer and start asking questions about. So we can download this data, and so we know how to read it. So cool. Have we solved challenge one? Well, sort of. We can read the data off, except that's not really the end of our challenge. It's just removing that bottleneck and pushing it down the line. So tell us a little bit more about that, Blake.
So this is kind of where we run out of the science world and run straight into the computational science world. So I work at a group with a group called Cyverse, and I am labeled the code-curious biologist. We have engineered ourselves out of the science, and now we've gotten squarely into the needs to visualize, move data, analyze data, and actually move it out into the greater whole of the world, past the ivory tower, like Sophia was talking about. My heart actually skipped a beat when she said ivory tower, because I've been saying that for years. So we're kind of running into bottlenecks, though, because our training as biologists is very traditional. We spend a lot of time learning about all the intricacies of biology, and I'm going to explain a little bit of that to you here today. But then when it comes time to actually move into these modern problems, we just get kicked to the curb, and people assume that we're smart enough to figure it out. So, to which end, I'm going to describe a little bit of biology to you. And my favorite unit in biology is the Harry Potter equivalent. Yes. I used to use War and Peace, and then people didn't know what that book was, so I've stopped using that one. But basically, a Harry Potter equivalent is all 7 books in the Harry Potter series, the original one anyway. If you take all of the letters that are in all 7 of those books, and you match those up to those letters we were talking about before, the A's, T's, G's, and C's of your genetic code, that's one Harry Potter equivalent. So it turns out that E. coli is almost exactly one Harry Potter equivalent. That is your gut microbes that help you eat, digest, and continue on. Yes, that's right. You are not just human. You're a walking ecosystem. But E. coli is kind of boring to me. I prefer this organism. This is yeast, man's oldest and best friend. It brings us wine, beer, and bread. Need I say more? And that's about 4 Harry Potter equivalents. So it's about 4 times bigger. The genome of that is about 4 times bigger. But then when you move into even higher organisms like insects, very, very small plant genomes, or nematodes, you get about 38 or so Harry Potter equivalents. So you can start to see this is ballooning out of size. And in fact, if you tried to read 36 or 38 Harry Potter equivalents, if you read every day, 7 days a week, 12 months a year, without sleeping, eating, or anything else, it'd take you about 8 years to do that, a little bit more. If you're a speed reader, maybe it'd only take you 5 years, right? So the data starts to balloon. And that's not even to mention humans, which stand at about 1,000 Harry Potter equivalents, so basically 2 orders of magnitude more than some of those simpler genomes. And the kicker there is that that's only 1/2 of your genome. So in reality, humans are complex, but what we talk about as a genome is actually just a small part. But when you actually take humans and compare them to plants, then we get really small. Sometimes things like wheat, which is one of the things that makes bread along with the yeast, right? It's got about 6,000 Harry Potter equivalents. And the largest genome that we know to date is Paris japonica. It's kind of a tiny little flower. To look at it, you wouldn't think much about it. But it has 50,000 Harry Potter equivalents. So it is massive. So this is one of those kind of universal jokes. The big things are often very small and vice versa. So like I was saying before, just to compound that problem, we're really only talking about half your genetics. No matter what creature you are, except for a few rare examples, you get half your genetics from your dad and half from your mom, right? Whatever that may be. And so really, when we're looking at humans, we all have about 6.5 billion, with a B, letters. And so that's about 2,128 nucleotides. Oh, but wait, it gets even worse. Because what we're really talking about there is those 2,000 Harry Potter equivalents is just a single cell. You have about 15,000 or 15 to 70 trillion cells in your body. And I have no earthly clue how to visualize this. But all of those genomes in your 70 trillion cells or so, plus all of the other creatures that are in you, that's not just human cells, right? All of your E. coli and your other commensals, all work together every moment of every day to continually respond to everything that you put your biology through, whether you treat it nicely, like I have not been, or you treat it nicely, right? And it constantly is working together. So we sat down because this is a visualization conference after all, and so I had no earthly clue how to visualize this. So I said, let's look elsewhere, right? So I said, let's look at all the stars visible in the universe, and I said, that's got to be trillions. Well, actually, it turns out to be 100 billion. Ascher thankfully pointed that out to me. So what you see here is all of the visible stars in a section of the sky, and if you actually take all of the stars you can see at night and in the universe, you got about 100 billion. And so we're not even to a trillion yet. And remember, at lowest, you've got about 15 trillion cells that all kind of are working together. And so now you really, and remember, each one of those stars is a cell, so you'll have those 6.5 billion nucleotides. Each of those are a data point. And it may seem like this is not that important, but each of your cells is incredibly important, because even if you get one cell that's a bad actor, if it gets a small change in just one cell, you've got what we call cancer. So when you actually ask scientists or medical people to go and find cancer inside your body, what you're literally asking is to take, 2 orders of magnitude more stars than you can see in the sky and find the one star that's bad, right? So there it is in the right corner there for everyone who hasn't found it, right? So you're identifying cancer in that one. So each of those cells is incredibly important. They all work together constantly. And if even one goes wrong, then you can have troubles. And that's inside those cells, right? So I'm talking about huge, but actually we're only considering the DNA in the nucleus. And that's not the only place you find DNA. So not only do you have, 70 trillion cells, they each have billions of nucleotides, we're actually only talking about the nuclear DNA. The mitochondrial and the chloroplast DNA, if you're a plant, no plants? Okay. That becomes even more complicated, and we don't even usually speak to those, and certainly not at this scale. So the data problem and the visualization and the analysis problem goes crazy. And speaking of visualization problem, that is not at all what a cell looks like. These are what cells look like. And you have thousands and thousands of mitochondria for every cell. Each of those have their own DNA. And if you're a plant, you have thousands and thousands and thousands of chloroplasts that do photosynthesis for you to create food. And those each have their own DNA, right? So to really sum it up, that's just genomes. Here's your textbook biology that everyone gets in high school or middle school or somewhere down the line. This is nearly so oversimplified that it's a complete lie. This is biology, right? DNA turns into RNA. RNA actually gets turned into proteins, which are like little micro machines that keep you alive and do all kinds of crazy things. And then they create chemistry and do a whole bunch of other things that actually keep you and make you who you are and create your physical body and all that other stuff. Can't speak to souls yet, but we're working on that, right? But they all interact with each other, and there's also a lot of outside forces that interact with each other. There's time, there's changes in structure of your DNA inside of each of your trillions of cells. There's the metabolites will actually affect the RNA, the proteins, and the DNA, etc. So it gets incredibly complex incredibly quickly, and you wind up asking these really deep philosophical questions at 3 o'clock in the morning when you're studying all this, right? And that is, what the heck is biology actually, right? We've oversimplified it in our minds so far that we can't really speak to it. So in these times, I like to remember those epic words as written by Douglas Adams in The Hitchhiker's Guide to the Galaxy, but as spoken by my spirit animal, Beaker, right? I even have the same hair. And that is, don't panic, right? Because even though there's a lot of chaos in biology, there's some very fascinating stories, there's some very, very interesting data to find, and there's a lot of ways that we can actually address some of the problem in the world. And so that's kind of what we do. Ascher works for Koji, which is a comparative genomics platform. I work for Cyverse, where we build cyber infrastructure for scientists. We started off doing it just for plant scientists, and now we do all science, right? And we're NSF funded, so we really do have an interest in empowering scientists to move out of their disciplines and get into computational science, visualization, and all the, how to move all this data, how to maintain all this data. But not only stop there, because again, we're NSF funded, we really have to move beyond that and get the education and training and outreach out to the greater whole. That meaning all the other humans on the planet. And if there's dogs that'll listen to me, I'd be happy to teach them too. So this is really what we're doing. So we're coming to you guys as end users and people who can deliver some of these technologies you guys have been talking about to actual scientists, to end users, to engineers, to astronomers, to whomever.
Oh yeah. So I hope he's convinced you that we have a lot of data. And I would like to now show you what our data looks like to us. Looks like that. It's a whole lot of that. A's, T's, G's, and C's. But I mean, it's pretty impressive that all of this, as simple as it is, can create things like this. So your body is a machine, just like any other machine. You have highways. They don't have cars. They have little tiny packages of cellular materials that are moving down them. And when you get out of it, you even have more. This is possibly my favorite side-by-side comparison I have. DARPA spent $100 million to build this robot that it's really impressive. It doesn't fall over if you push it.
Meanwhile, humans can do backflips and all kinds of things. So, the engineering world really has ways to keep up with just these simple biomachines, these proteins that you have. But even more interestingly, when we were talking before about biotech, there's ways to actually move these machines around. So many years ago, people got really interested in spider silk. And instead of raising spiders on a farm, because not only is that creepy, but it's also kind of scary and silly, people have actually taken the genes that make spider silk, a material that's actually harder than steel, lighter than a feather, and have put them into goats. And that's what you're seeing on the left. So you milk the goats. They have spider silk in their milk, and you pull the spider silk out, and you can do all kinds of crazy things, like weave it into Kevlar vests that'll stop bullets. There was actually a group of scientists and an artist, lovely interactions that I love to see between domains. They actually put the spider silk into human cell cultures and created a layer, and now you have human cells that stop bullets, right? So you get a lot of really fascinating applications in addition to some of the other things we've been talking about, right? And again, all of that is in there somewhere. It's just really a matter of finding it. And because it's just A's, T's, and G's, A's, T's, G's, and C's, it's really hard to do that. And so this is where we get into our challenge of how do we actually understand all of this so that we can do these applications, we can teach people, we can start to do good science, right? And so really what we have to do is we have to take all that raw data and transform it into something that's human readable, distributable, reproducible, and all these things. So what you're seeing here is assessments of genes and gene placements. You'll notice very quickly that the whole genome is not actually turned into proteins, those micro machines that actually go out and do things. They're kind of like islands, and you have to go hunting around for them, and that's what you're seeing in these bars here. These are all individual genes, and then you can actually stack up a lot of different kind of data types to inform your information. Now, we've come very far in a very short amount of time, but we have a lot farther to go. Once upon a time, when we first started doing genetics, we would actually take all those A's, T's, G's, and C's and write them on the top of a blackboard, and then match up other sections of A's, T's, G's, and C's on the bottom of the blackboard and draw lines between them. Not very high throughput, as you may imagine. There was algorithms, so that's when biology first turned to computational science, and so algorithms were designed to actually do alignments, like what I was talking about, taking two pieces and actually matching them up. And you started getting blasts and other types of visualizations like this here. So now we can actually compare genetic sequences between organisms or pieces of genetics between organisms. Expression data across the whole of a genome. You actually have different parts of that genome getting turned into different things. And so you can actually, from some of these basic visualizations, you start to realize there are these hot spots and then there are these cold spots. This one is my particular shame. This is something that's been existing inside of the genetic world for a very long time. We call them heat maps. Now, why they're called heat maps is an interesting historical story, and I'd be happy to tell you, but heat usually means red and blue to me. It also just so happens that red-green color blindness is the most common color blindness, especially prevalent in males, which regrettably also happens to be the majority of a lot of these sciences. So not only did they create visualizations that are based off of bad colorblind visuals, but they also did it to themselves, which is kind of funny when you think about it. This is a karyotype in the bottom left. So they've actually extracted all the chromosomes, all of the genetics, all the genome out of a single cell and squashed them and visualized them. So each of these chromosomes, one you get from your dad and one you get from your mom. You are exactly half-half. One half of your dad, one half of your mom. You may not like that, but that's just how it works, right? These are syntenic dot plots, and so you can see we're starting to match up genetics across different organisms and see what is similar, what has been inherited, what sections have passed down over heritable time. And so you start to get into some of these complex visualizations. These have been static in the past, and that becomes problematic when you're trying to understand all of these interconnection points, especially across 6.5 billion nucleotides. And then, of course, you have the ever-famous Circos plots that can allow you to actually stack up all kinds of different types of data, DNA, RNA, methylation data from epigenetics, et cetera. And you get a lot of kind of fascinating interactions there. And so we really are starting to move in the right direction, but we have a lot farther to go.
So Blake had mentioned that I work for, and he previously worked for, a comparative genomics platform. So I wanted to take just a minute to sort of talk about what we've been working on within our confines to make this problem easier, make understanding more accessible to scientists that might not be able to use complex tools. And so our whole thing is web-based, and I'm going to just try to explain to you without turning around so you can hear me what's going on, and then I'll start playing it. So on the side that's playing right now, this is a wonderful tool that my advisor made when he was a graduate student. And what you do is you can see on the other video, you have an XY axis, and what we've done is just stretch those genomes out. So you have genome 1 on the X, genome 2 on the Y, and then we draw a dot everywhere those genomes are the same. So if you would imagine you were to compare a human and a human, you would wind up with a diagonal line. Here we have corn and sorghum, which are still pretty similar. You can see very large chunks of that are the same. And so this wonderful program that my advisor made, it's very advanced. It runs some calculations on the server side. It generates a static PNG that it returns to you. You can click on that. It then goes back to the server and says, hey, we want just this section. Run some more calculations, generates another static PNG. But that's not really super good, especially if you're interested in looking at smaller areas. How does that work? So what me and Sean Stevens, who's not here, have worked on is actually converting this into an interactive version using D3 and cross-filter and tools like that, so that you can actually start to just ask questions about individual parts of it. And you can look on the histograms, which display some other information about how genomes work, and start to brush across it and see how these things are working together, look at just areas you're interested in. And it has been pretty well received by our end users, who are now able to ask a lot more detailed questions than they were before. And so that was a pretty good improvement. And here's another one. So this is a really interesting phenomenon in genomes, is actually sometimes a whole genome can just copy itself, and now you have 2 copies of everything. And that's pretty cool, but not very good. So the cell will try to, or not the cell, but the whole organism will try to eat it down to where it has just this double state again, but this doesn't happen uniformly. Certain parts are lost, certain parts are retained. So what we do is we do an analysis to look at, when you look at 2 organisms, where have things been lost versus retained, and we get these nice line plots. So this was the first version of the tool on your right-hand side. It was using matplotlib. Some pretty nice, you can see on the top, pretty nice plots, pretty clear, but this starts to break down when you get into species that have more chromosomes or more similarity, and on the bottom you can see that's just not gonna work anymore. Those are too crazy. Too many lines, you have no idea what's going on. You have pinks, you have 4 different shades of pink in them. Very difficult to comprehend what's happening. So thanks to Plotly.js, we were able to make these interactive, so you can start to go in, turn those on and off, zoom into just the areas you're interested in, and overall get a better idea of what's happening in those genomes, where you're interested, and use that to make interesting discoveries about what's happening in this process. And so this is actually probably my other favorite part about our platform, is that we're really interested in reproducibility. So all of our pipelines that are analysis pipelines, we make available at every step, every raw data file, so we're not a black box. When your data goes in and is transformed into results for you, you can go back and see every step that's happened. You can download those, and if you start to question the validity, you have the ability to go in, look at it, and figure out where something might have gone wrong. And we always generate a link at the bottom so that you can send that along with your publication and say, hey, if you don't believe me and you want to try this analysis yourself, go to this link, and that's it. So here's our visions for the future. I believe we're actually out of time, so you can just read them. I guess the most important one is we're always trying to move towards multi-dimensional visualization. So if anyone has thoughts on how to visualize 10,000 things compared against 10,000 other things, I would love to talk to you, so please come find me and we can discuss that. So with that, we'll take a question if we have time. We don't have time.